Welcome back to Science Lullaby. Tonight, we'll take a calm journey into the meaning of life itself and discover why every living thing, no matter how different it may appear, is connected by something deeply fundamental. The surface of our planet is filled with life, curious, intricate factories of chemistry, each one capable of taking in matter from its surroundings and quietly turning it into copies of itself. When we look at the living world, the variety seems endless. What could be more different than a tiger and a piece of seaweed, or a tiny bacterium and a tall tree? And yet, our ancestors, long before they knew of cells or DNA, recognized that all of these things shared something in common. They called that something life. They marveled at it, they tried to define it, and often despaired of ever explaining it. In the last century, discoveries have changed how we see life. Instead of removing the mystery, they've only deepened the wonder. But one central puzzle has been solved. We now know that every living thing is made of cells. Cells are small, membrane-bound units filled with a watery solution of chemicals, each one with the extraordinary ability to grow and then to divide, creating new copies of itself. Because of this, cells are often called the fundamental units of life. And it is to the study of cells, cell biology, that we must turn to answer the most basic questions of existence. With a deeper understanding of cells and how they evolved, we can begin to approach the grand mysteries of life on Earth, its mysterious origins, its incredible diversity, and its ability to occupy every habitat we know. As one early biologist, E.B. Wilson, once said, "The key to every biological problem must finally be sought in the cell. For every living organism is, or at some time has been, a cell. And despite the dazzling variety of living things inside, they are surprisingly similar." This is one of the great themes of biology. On the one hand, astonishing variety in outward forms, and on the other, astonishing constancy in the basic mechanisms that keep them alive. It is estimated that there are more than 10 million, perhaps even 100 million species of living organisms on Earth today. Each species is unique, and yet each reproduces itself faithfully, handing down detailed instructions from one generation to the next. This is heredity, the passing of information from parent to offspring. It is what makes life different from other orderly patterns in nature, like a crystal growing, or waves forming on the ocean. Yes, a crystal can grow and a wave can move beautifully, but neither carries instructions that connect one generation to the next. Living things do. Like the flame of a candle, every organism must consume free energy to keep itself going. But unlike a candle, life uses that energy to drive an astonishingly complex system of chemical processes, all guided by information passed down from the cells that came before. Most living things are made of just one cell. Others, like us, are vast collectives of cells, entire cities of them, working together, each with specialized roles, connected by intricate systems of communication. Even so, the human body, with its more than 10 trillion cells, began from just a single cell. That single cell carried all the instructions needed to build a body. It had the machinery to gather raw materials from its surroundings and to use them to construct a complete copy of itself, right down to its hereditary information. Each and every cell is truly extraordinary, small beyond imagination, but powerful enough to carry the story of life forward. And so, all life, from the tiniest bacterium to the tallest tree, traces back to one remarkable truth. Every living thing, no matter how complex, is built from cells that share the same universal blueprint. These are the universal features of life on Earth. Each cell is wrapped in a delicate membrane, a boundary that separates it from the chaos outside, yet allows a quiet conversation with its surroundings, a constant exchange of energy and matter. Inside, a shimmering dance of molecules unfolds. DNA stores the instructions of life, written in a language shared by every organism that has ever lived. Proteins carry out those instructions with purpose, building, repairing, sensing, and transforming. And all of this happens in water, the cradle of chemistry that sustains every living cell. Whether it belongs to a blade of grass, a blue whale, or a human being, every cell follows the same ancient rules. They all take in energy, convert it, and use it to maintain their delicate order. They all grow, they all divide, they all pass on their information to the next generation, keeping life's flame alive, unbroken for billions of years. Even though life's shapes and colors seem endlessly diverse, deep inside they are kin, bound by the same invisible thread that connects every cell on Earth. Every cell, no matter how simple or complex, speaks the same ancient language of life. It stores its hereditary information in a single, elegant code. DNA, deoxyribonucleic acid, a name that may sound heavy, yet what it carries is the essence of every living being. In the digital world, we've grown used to the idea of information. Bytes, bits, codes that store our words, music, and images. Life, too, is an information system, one that has been running flawlessly for over 3.5 billion years. Every living cell, from a bacterium floating in the sea to the neurons inside your brain, uses the same molecular archive. Two intertwined strands, a double helix, each made of smaller building blocks called nucleotides. They come in 4 types, marked by the letters A, T, C, and G. These 4 letters form life's alphabet. Their sequence, their precise order, is what encodes every trait, every function, every whisper of biological design. What's extraordinary is that this code is universal. You can take a piece of human deoxyribonucleic acid and place it inside a bacterium, and the cell will read it perfectly. Or Insert a gene from a bacterium into a human cell, and it will understand. Across all of life, the script is written in the same language. Scientists have learned to read this script, letter by letter, uncovering the complete sequence of DNA that defines each organism. Millions, sometimes billions of nucleotides, forming a story so long it could fill libraries. And yet, it is written using only 4 letters. DNA is not just a molecule, it is the memory of the planet, the continuous thread that connects every living cell to the very first spark of life. Keep life going, every cell must perform one essential act. It must copy itself, not just in shape or motion, but in memory. Every living thing carries a record of what it is, written in the precise order of its DNA. When a cell divides, that record must be passed on. And so begins one of the most elegant dances in biology, templated polymerization, the process by which DNA makes a faithful copy of itself. Each strand of DNA is made of repeating units called nucleotides, tiny building blocks made of a sugar, a phosphate, and one of 4 bases: adenine, thymine, cytosine, and guanine, or simply A, T, C, and G. These bases are the letters of life's alphabet. A pairs only with T, and C pairs only with G. A perfect system of complementarity. The 2 strands of DNA twist gently around each other in a double helix, like intertwined ribbons of information. When it's time to replicate, the 2 strands are pulled apart. Each becomes a template, guiding the construction of a new complementary strand. A on one side calls for a T on the other. C invites G. Base by base, the code is copied perfectly until 2 identical double helices emerge. The bonds that hold the strands together are delicate, allowing them to separate easily, while the backbone, made of strong sugar-phosphate links, keeps the structure intact. It's a design both sturdy and flexible, one that has preserved life's message for billions of years. No matter the organism, a bacterium, a tree, or a human being, this same process unfolds within every living cell. DNA replication through templated polymerization is universal. It is how life remembers. It is how life continues. DNA does more than just make copies of itself. To bring life to motion, it must express its information, turning silent code into living chemistry. This expression follows a universal rhythm shared by every organism on Earth. It begins with a process called transcription, where DNA's message is copied not into another DNA strand, but into a closely related molecule, RNA, ribonucleic acid. You can think of DNA as the library and RNA as a messenger carrying a single page of its instructions out into the bustling city of the cell. RNA looks much like DNA, but with small, elegant differences. Its backbone uses a sugar called ribose instead of deoxyribose. And instead of the base thymine, RNA uses uracil or U. Still, the other letters, A, C, and G, remain the same. During transcription, one strand of DNA serves as a template. RNA nucleotides line up in perfect complement. A pairs with U, T pairs with A, C pairs with G, and G pairs with C. Each match adds a letter to a new RNA chain, a short copy of the information stored deep within the genome. Unlike DNA, which stays carefully protected inside the cell's nucleus, ribonucleic acid molecules are temporary, made, used, and then replaced. They carry messages, coordinate reactions, and guide the making of proteins. They are the working notes of life, flexible, disposable, but essential. Some RNA molecules act as messengers called mRNA, delivering genetic instructions to the cell's protein factories. Others fold into intricate shapes, forming structures that help decode the genetic message, and some, astonishingly, can even act like enzymes, speeding up chemical reactions on their own. This remarkable versatility is why many scientists believe that life may have begun with RNA, a molecule that could both store information and perform chemistry, a single strand capable of both memory and action. Even today, in every living cell, that story continues. DNA holds the archive, RNA carries the message, and together they bridge the gap between information and life itself. Inside every living cell, the quiet code of DNA comes to life through one extraordinary class of molecules, proteins. Like DNA and RNA, proteins are long chains built from repeating units, but instead of 4 letters, they use 20. Each of these building blocks is an amino acid sharing a common backbone yet carrying its own unique side group. A chemical personality that gives every amino acid its distinct flavor. Strung together in a precise order, these amino acids form a polypeptide chain, a sequence chosen and refined over billions of years of evolution. But what makes proteins truly remarkable is what happens next. As the chain folds, it twists and curls into intricate 3-dimensional shapes, each one designed to do something specific. Some form the cell's framework, giving shape and structure. Others generate motion, transport nutrients, or sense signals from the environment. But above all, many proteins become enzymes, catalysts that make life's chemistry run. An enzyme can speed up reactions that would otherwise take centuries, making or breaking bonds in the blink of an eye. Every heartbeat, every breath, every thought you've ever had depends on countless enzymes working in perfect coordination. Together, proteins carry out the vast majority of the cell's work. They build, repair, and regulate, turning genetic information into action. It's a remarkable feedback loop. DNA holds the instructions to make proteins. Proteins, in turn, build the machinery that copies DNA. And through this cycle, this elegant molecular dialogue, life sustains itself, generation after generation. At its deepest level, a cell is a self-replicating collection of catalysts, a living chemistry set that takes in energy and matter, uses them to make more of itself, and lets the leftovers go. That is what life is, a dance of molecules driven by information, guided by chemistry, and sustained by time. Once the message of DNA has been transcribed into RNA, the next step begins, one of life's most elegant acts of translation. Every cell on Earth follows the same remarkable code, from the simplest bacterium to the human brain. The way RNA is translated into protein is nearly identical. In this process, the four-letter alphabet of RNA, A, U, C, and G, is read 3 letters at a time. Each triplet, called a codon, corresponds to one of the 20 amino acids that build a protein. 4 letters grouped in 3s. That means 64 possible codons, enough to encode all 20 amino acids with room to spare. This code, the genetic code, is universal. It's been passed down from the earliest life forms, its patterns so deeply woven into biology that any change would be catastrophic. What began as a frozen accident in life's earliest history became the foundation of every living cell that followed. The decoding of this language happens on a magnificent molecular machine, the ribosome. Here, small molecules called transfer RNAs, or tRNS, act as interpreters between the languages of nucleotides and amino acids. Each tRNA carries a specific amino acid at one end and a 3-letter anticodon at the other, a mirror sequence that pairs precisely with its matching codon on the messenger RNA. As the ribosome moves along the mRNA strand, codon by codon, the TRNAs bring in their cargoes, amino acids, one by one. Each is linked to the next, forming a growing chain, a protein taking shape, letter by letter, bond by bond. This is how the information once stored silently in DNA is finally transformed into motion, structure, and life itself. The ribosome reads, the TRNAs deliver, the amino acids join, and from chemistry, form emerges across billions of years, across all species. This same translation occurs. It is the shared language of life, one that speaks in triplets and builds the universe within us, one protein at a time. Within the vast molecule of DNA lies the blueprint of every living thing. But this blueprint isn't one continuous scroll. It's a library of thousands of separate instructions, each one devoted to a specific task. Each instruction is called a gene. A gene is a segment of DNA that carries the information to make one protein, or sometimes several alternative versions of it. Special sequences within the DNA act like punctuation marks, telling the cell where each message begins and where it must end. When a gene is read, it produces a ribonucleic acid copy, a messenger molecule that carries that gene's instructions into action. And from that message, the corresponding protein is built. But nature is not rigid, it's creative. In more complex organisms, a single gene can be processed in multiple ways, producing slightly different forms of a protein, each with its own role. It's as if one paragraph of DNA could be read with different pauses or accents, changing the meaning while keeping the same words. Not all genes make proteins. Some give rise to RNA molecules that never leave the world of nucleic acids, RNAs that regulate, stabilize, or even catalyze reactions within the cell. These, too, are genes, part of the same genomic orchestra. Still, the cell doesn't play every note at once. Each gene is regulated, switched on or off, depending on what the cell needs. This control comes from additional DNA sequences, regulatory regions, places where special proteins bind to decide whether a gene should be active or silent. This regulation is the reason a neuron and a muscle cell can share the same DNA, yet look, behave, and function completely differently. Each cell reads only the genes it needs, turning the genome into a dynamic living script. So the genome, the full collection of all DNA, doesn't just define what a cell is, but also when, where, and how each of its parts should come alive. In this symphony of molecules, genes are the notes, proteins are the instruments, and life is the music. Every living cell is a tiny chemical universe, restless, organized, and full of motion. Unlike the stillness of a crystal or a rock, a cell lives far from equilibrium. To stay alive, it must constantly take in free energy from its surroundings. This energy fuels the reactions that build and repair, that copy and divide. When that flow of energy stops, the order collapses. The molecules drift back toward equilibrium, and the cell dies. Life, in its deepest sense, is the fight against disorder, the art of maintaining structure in a world that tends toward chaos. Free energy is the currency that makes this possible. It powers every heartbeat, every leaf unfurling, every strand of DNA being copied. Without it, even the instructions of life, its genetic code, could not be written or preserved. Imagine the molecules inside a cell as countless particles in motion, vibrating, colliding, tumbling randomly in thermal chaos. To build something meaningful from that chaos, a strand of DNA, a membrane, a protein, the cell must capture, organize those molecules in a precise order. That ordering takes energy. When two molecules find their proper partners, they bind, and in doing so, they release a small burst of energy, like a spring settling into place. Each bond formed, each pattern stabilized, each instruction recorded, all are acts of order born from the expenditure of free energy. To replicate its DNA, to grow, or simply to survive, the cell must continually draw that energy from somewhere. For animals, it comes from the chemical bonds in the food they consume, energy once captured from sunlight by plants. And for plants themselves, it comes directly from the light, harvested by chlorophyll, converted into the fuel of life. In this endless exchange, sunlight to sugar, sugar to motion, motion to heat, the same rule holds true across all of biology. Life persists by spending energy to create order. And when that energy flow stops, order fades into stillness. Every living cell is a tiny biochemical factory, endlessly busy, quietly transforming matter and energy into the stuff of life. Inside, billions of reactions unfold every second. Sugars are broken down, proteins are assembled, and new DNA strands are woven from molecular threads. No matter the organism, a bacterium, a tree, or a human being, all cells work with the same basic set of molecular building blocks. They all handle sugars, nucleotides, amino acids, and lipids, and they all rely on a single universal molecule of energy, ATP, adenosine triphosphate. ATP is both a building block and a battery, carrying packets of free energy that power nearly every chemical process inside the cell. But cells differ in how they gather what they need. Plants can take sunlight, water, and a few simple nutrients, and from them build everything. Animals, on the other hand, must consume other living things to obtain many of their complex molecules ready-made. Yet, whether through photosynthesis or digestion, all cells ultimately run on the same chemistry of energy and order. Every cell is also surrounded by a delicate boundary, the plasma membrane. This thin, flexible film keeps the cell's inner world distinct from the chaos outside. It allows nutrients to come in, waste to go out, and precious molecules to stay protected within. The secret of this barrier lies in its structure. The membrane is built from amphiphilic molecules, compounds with one end that loves water and another that avoids it. When placed in water, these molecules self-assemble into a double layer, their hydrophobic tails tucked safely inside, their hydrophilic heads facing out. This simple behavior, guided by physics and chemistry alone, forms the membrane that makes life possible. Yet a cell cannot live behind a wall. The membrane is selective, not sealed. Specialized transport proteins act as gateways, letting in nutrients and exporting waste. These transporters are so ancient that scientists can still recognize their shared ancestry across bacteria, plants, and animals alike. The proteins within the membrane decide what may enter. The enzymes inside decide what happens next. Together, they define the cell's chemistry, its metabolism, its structure, its behavior. All of it flows from the information written in the DNA. But how much information does it truly take to make a living cell? Surprisingly little. Some of the simplest known organisms, like Mycoplasma genitalium, live with barely 530 genes, only about 400 essential for survival. Its entire genome, just over 500,000 DNA base pairs, contains about as much information as one short chapter in a book. And yet, within that modest code lies everything it needs to grow, divide, and pass on life to the next generation. Even the smallest cell is a masterpiece of efficiency, a self-replicating system powered by energy, bounded by a membrane, and guided by the same molecular principles that govern us all. Life may be complex, but at its heart it follows elegant, universal rules, a single language of molecules, spoken by every living thing on Earth.